So last talk before the break. Let me just see where I'm going to stand here. So this is really following the spirit of Zoltan's talk. And I know when people hear the word parapsychology, it makes them shiver. Most of you, I can see that. Is ESP real? Psychokinesis, telepathy, all those things. But here we are at Stanford, and one of the first controlled psi experiments happened here in 1911. I'm going to talk about that. And so we have a 100-year history. But what I'm really going to talk about is a new protocol in that context that is being tested in the context of a well-known psi study. There's another one. This was the BEM 2011 study, but there's also another study of Dean Radin with a series of publications over 10 years now, even preceding publications were starting in 2012 again until now, claiming a psychokinesis effect in a particular system. So this is, again, not endorsing, not denying. This is really about a test case. How can you improve protocols to get to really the bottom of what's going on? Not in terms of mechanisms, but in terms of capturing better measurement errors in a system. And that's the technique of counterfactual meta-experimentation. So just briefly, clearly, given the current research reform movement, parapsychology will be re-examined. There's lots of people there, lots of laboratories still doing work. Two possible outcomes. One, the proponents will argue sometime credible, true positive data will be produced. The skeptics clearly say what will be shown is that parapsychology is all about uncontrolled false positive results. And I'm not taking either position here. Could any of the published results be true positives? We don't know yet. But the key question we've engaged in with our work was how to empirically distinguish true positive from false positives in the same experimental system while performing the same study. And I should say I'm now director of the Fetzer Franklin Fund, as John mentioned. But the first 15 years of my scientific career, I was an experimentalist in the laboratory. And actually, with the help of Fetzer at the time, we founded the Bioelectromagnetics Laboratory in 1994 at Stanford University, which I was running for about 8 years as a director, assembling a team of 7 scientists to study the effects of electromagnetic fields on living cells, on the human body. Remember in the 90s, the whole electrosmog debate, do power lines cause cancer? Now we have the debate about, does our iPhone cause brain cancers? So it's all about these very subtle influences, electromagnetic influences in this case, and whether they might have any effects. Who was it this morning talking about, the magnet study, the New York Times study? Do magnets help in pain? These sort of questions. And we studied them for 8 years in the laboratory and developed many protocols of different kinds to actually see, is there anything to that? Can there be true positive data? What kind of false positive effects can occur? Are there models for it? So we've done combined experimentation with computer simulation and theoretical modeling in this very small team. And that was kind of unique. We hired at that time electrical engineers, theoretical physicists, cell biologists, all together working in a group for many, many years. Out of this came these ideas for these protocols I'm going to present to you that we now applied in a large-scale confirmatory replication trial on this experiment. But here, since I'm speaking here also as a director of the Fetzer Franklin Fund, I also want to take the opportunity of looking at the limits of science and what meta-science is. Before I do that, let me just explain to you, what does this strange term mean? Counterfactual meta-experimentation. These are experiments testing the performance of the experimental method itself in the absence of any experimental or control condition, hence the term counterfactual meta-experiments. So these are sham experiments on a system, very simple idea, very rarely applied in a systematic fashion, but extremely powerful in uncovering false positives that might be hidden somewhere in your experimental design. What do I mean by parapsychology as a control group? Obviously, this is the skeptical position. This is just meaning in the sense that if psi does not exist, the experimental condition should be equal to the control condition. End of story. So what about the limits of science? I'm just going to spend 3 minutes on the frontiers of meta-science that we are not discussing at this meeting today. But we are very interested at the Fetzer Franklin Fund and also among the organizers for a meta-science symposium that might happen in the future. So even the question, what is science? Theories of science. What metaphysical assumptions underpin modern science? At present, clearly, it's scientific physicalism or materialism. Is scientific physicalism incomplete as we understand it today? Might a future science include the scientific observer, agency, consciousness in its frame? Because right now, science cannot even account for the scientist. All we can do is account for the operations that are going on in the laboratory. But we tried to get at that on the first day in the morning. How do scientists generate ideas? How research experiments are done? But until we understand what the nature of consciousness is, we cannot even understand what the scientific process is. So it's a big blind spot, I think, in the discussion, and that's certainly a meta-scientific frontier topic in the future. Remember, if you look up meta-science, what the definition is, there's really two definitions. Meta-science is the foundations of science or philosophy of science, the foundational principles of science, and meta-science as the science of science, the practice of science in studying how science operates. And these are complementary. In this meeting, we really focus on the aspect of the operations of science. But one really interesting question is, for a future meta-scientific inquiry, are the foundations of science fixed, or are they future for open revision? What might go on in 200 years with science? We don't know that. We're just making assumptions right now. So what makes possible scientific truth and objectivity is another one of these questions. Is the reality that science describes absolute or always model-dependent? What are inherent limitations to scientific understanding? How to differentiate science from pseudoscience, the demarcation problem? Could phenomena exist in the world that elude capture by present scientific methodologies? What should be their status as we talk about these, like the ESP case that Zoltan just talked about? What is the relation between the scientific discovery process and the resulting scientific image of reality that's being created in the world that we act upon and do our science by? What is the role of the adopted worldview of the scientist in the conduct of science, interpretation of evidence? That's a question we already have been starting to get at this particular meeting. Most importantly, are the limits of science the limits of scientific reproducibility? How to distinguish laboratory observations that are true positive, but inherently unpredictable and non-reproducible? How to distinguish those from uncontrolled false positives? When you look at quantum theory, quantum experimentation, the only thing that's reproducible is the non-reproducibility of individual outcomes. So all these things we're not addressing, and I think there's a vastly bigger scientific framework that we need to look at in the future, beyond the practical operational aspects of the things that we do. So what are the fundamental limits of science versus what are the practical limits of science? What is the future of scientific explanation? Will the meaning of explanation change over time? Certainly it has in the history of science, from causal explanations to operational explanations, for example. Can existing limits be overcome by new advances in meta-science? And finally, this is the question I'm going to talk about in this example experiment that we've done over the last years, how to empirically distinguish false positive from true positive data in a single experiment. Granted, large-scale experiment to do this. So this symposium is mostly focusing on these practical aspects of the process, and I think that's where we need to start. I think there's a great need, as we've seen, to reform the research, research. But I think we should not lose sight of the other questions that are the foundational questions about the very nature of science and what makes science work. So just briefly, 100 years of parapsychology reforming research. So did parapsychology reform research? No. Or does parapsychology require a reform of its own research process? Clearly. But this is what I want to talk about. This was the first major psi study. I don't know if many of you know that. This happened at Stanford University in 1911 by Dr. Coover, who was a psychologist. And he was funded by the brother of Leland Stanford, the founder of Stanford University, to run 10,000 test trials on ESP, just like the BEM study already exactly 100 years before the BEM study. That might be a coincidence. That's why I'm calling it 100 years of parapsychology. Then, of course, J.B. Ryan set up a lab at Duke University. A journal was formed. The Parapsychological Association became an affiliated member of AAAS, It is until this day, by the way. Many of you might not know that. Then, of course, Stanford Research Institute, now SRI International, in Menlo Park, nearby here, carried out research in parapsychology, the leading work in this time, during this time period. The leading scientists were Harold Puthoff and Russell Park. And actually, Nature, in 1974, published, I don't know if it's the only one, but a side paper on information transmission under conditions of sensory shielding, on remote viewing, written by Harold Puthoff and Russell Park. And actually, Harold Puthoff is today here in the audience. So if you want to talk to him about what he thinks, what the status of psi research is today, please, I don't know where you are. Hal, are you here? Okay, please here. One of the leading researchers in this field to this day. Then Princeton University had a major anomalies research center on psychokinetic effects on random event generators. And at the present time, there's at least 30 research groups at academic centers throughout the world, at university settings, carrying out parapsychological research programs. At least 20 research groups that are privately funded outside that, and at present day, at least 15 journals are publishing psi findings on a routine basis today. So something is diverging here. You know, there is a whole community. So there's an epistemic, I think I loved Steve Goodman's, conversation. There's a whole social aspect to this. People have an epistemic, they operate in a completely different assumptions about, what is real and what isn't, and what is worthy exploring and what isn't. But I just wanted to point it out as a context that this is going on, and this has been going on for a long time. And there's thousands of peer-reviewed articles in the literature claiming evidence for psi effects. So to me, it seems this is a perfect, whichever way you take, either you're a skeptic or a proponent, a perfect case study of either what can go wrong or could go right in science. Because the predefined definition is psi effects do not exist. So is this all artifact? And if it is, we better find out how this could happen. And what went wrong with the scientific process? Maybe not, but maybe yes. So that's what I mean by as a control group. And there are these publications. And so the task, the skeptic would say, the task of the current research reform movement is to demonstrate how uncontrolled false positives can be produced for a given parapsychological research design. It's not enough to say, in my mind, oh, the effect can be replicated or not. I think advancement in knowledge will come if we can actually show how it happened, how a false positive happened in the system. And this was the basic idea for our protocol, which we call the Advanced Meta-Experimental Protocol. And it's really based on the question how to reveal uncontrolled false positives in scientific experiments in general. It's a universal protocol. It introduces new systematic error categories that this protocol can directly measure as you are performing the protocol in conjunction with your search for true positive results. So it includes, as its foundation, counterfactual meta-experimentation. And I claim it's a powerful research tool for detecting the hidden presence of false positive effects if they do exist in any experimental paradigm. This can be adopted to your experiment. So look at it through this lens, but it's been applied to replicating a psi study. In particular, for experiments seeking to reliably detect very weak or ultra-weak effect sizes, which, in physics, but also some studies in psychology are at this limit. The BEM study, certainly, that we just heard about, has percent effect size of 3%, 4%, I think. So I would hope that this protocol could be included at some point in Zoltan's study that he's doing. Now, this was just published. This is the work done in collaboration with Nikolaus von Stillfried, who is here, who presented a poster yesterday at the session. He's a senior research scientist at Phenoscience Laboratories in Berlin. So I'm in a fortunate position to be director of the Fetzer Franken Fund, to fund programs, to develop program areas, but I'm also still able to do my own scientific work, and mostly now on philosophy of science and interpretations of quantum theory, things like that, but also on developing novel protocols and actually testing them in the laboratory. So the AMP, I call it the AMP. So, as I say, amplify your research design by using the AMP, that would be a slogan, is implemented because false positive effects may still be generating distort the validity of findings, even with strictly confirmatory study designs, i.e. pre-registration, and even when technically accurate statistical analyses are performed. So there's still the danger, of course, of false positives. The reason may be, and as we say, is that false positive due to class A and class B error, and you've never heard about these terms. I just made them up as part of our study. I'm going to tell you what they are. May be indistinguishable from potential true positive effects. And just as an example for hidden systematic non-random influence, which we have termed class A error on the experimental outcome, think of a physically unbalanced or biased roulette wheel. So if you want to do a study, whether and people have done this, whether a psychic can influence a roulette wheel and to make more money, but you're using a wheel that is slightly unbalanced, so there's a higher probability for the ball hitting one of the octants predictably, even to a very small degree. If you run a large number of trials, you are guaranteed to make money. And you might think, oh, it's the psi energy of the person, but it's an unbalanced roulette wheel. This problem, this bias, cannot be accounted for by any pre-registration, cannot be accounted by any statistics. It's a totally different class of errors that you cannot address unless you actually directly measure this error. And that's what we've done. So what is class A error? It's a method-dependent systematic error, the type of systematic error that is associated with the scientific process itself, independent of the application of either test condition or control condition. Class B error is condition-dependent systematic error. The type of error is a function of the biased or unbalanced application of X and O conditions, X for experimental intervention, O for the control condition. The purpose of this new protocol, the is to quantify the amount of Class A error and Class B error of an experimental design. So there's three distinct test categories that are used in this. The experimental condition that we're all familiar with, the control condition that we've, but a third condition, which is called the sham condition. And the counterfactual sham experiment performs the experiment in the absence of the source of X and O, and that will be the sham condition. Think of an example in a clinical trial, or you want to test the effectiveness of aspirin on headaches. You give an active pill, and of course, you must use as a control condition a placebo pill. And what would be the sham condition? Would be the no pill condition. So you actually have three different conditions to test. So this gives you a toolkit for advanced research design, because you can combine all these conditions in many different ways to detect very different kind of errors that normally are not measured in experiments. So, for example, this is your standard experimental condition, X versus O, experiment versus control, but you could run a systematic negative control experiment of the control condition versus the control condition, or the systematic positives, experimental condition against experimental condition. You could run the control condition against the sham condition and actually measure what is the effect of my control condition on the system, which is rarely ever done. You can run, and then finally you can run a sham condition against the sham condition, which is the complete counterfactual experiment. So this is the standard experiment. This will be the counterfactual meta-experiment. And what is a counterfactual meta-experiment? The sham, we call it sham experiment. This is a long word, so let's just use the word sham experiment. It addresses these questions. What would have been the change in measurement outcome Y if the source that delivers the experimental and the control condition would not have actually been present in your system? So by mimicking as closely as possible the concrete experimental situation in the absence of the source of X and O, the counterfactual meta-experiment tests empirically the counterfactual prediction if X had not been present, my test intervention like ESP or a test subject, then the statistical effect on Y would not have occurred. So in summary, these sham experiments will determine the true negative detection rate, i.e. the specificity for X, in the language of statistical measurement theory of an employed methodology are done for one purpose only, to reduce the risk that a statistical XY correlation represents a false positive measurement correlation. So this is from the paper. This is statistics 101. Usually we don't measure this error. So what I'm saying is the sham experiment actually determines the specificity of the used methodology by quantifying the true negative detection rate, i.e. specificity. It's a logical thing to do, when you do mission-critical experiments. Certainly all psi investigators or all people who are working on extraordinary claims and they need to produce extraordinary evidence should just use a very basic application of this methodology. So this is the class A, this is the setup for the class A error detection. These errors that are associated with these conditions would be class B errors. So I'm going to switch now to explain these things on a concrete experiment that we've done over the last years. And then I will exactly explain what these errors are measuring. And the experiment is complete now. The results are published. Actually, they were published 2 weeks ago, this August. And so it's going to show you this, how to implement this protocol on a concrete case of this study. So the results claimed by the Radin experiment, very much like what Zoltan talked about with Daryl Bem. This is, in this case, not a precognition. This is a psychokinetic study. Can the mind influence matter, basically? And under controlled laboratory conditions. So test subjects there interact psychophysically with laser light waves interfering in a double-slit apparatus. Term would be psychokinesis to use for that. The Radin experiment suggests a remarkable skill. It claims to detect ultra-weak effects, systematic effects, and on the order of thousands of a percent. These are extremely large data samples, therefore, to achieve statistical significance. I mean, one typical experiment takes 80 gigabytes of data, just recording. So these are 10,000 test trials, individual test trials that are done in these cases. So these are these publications. So this is, even University of California is involved, a representative of that in the very early paper. There's other papers. So in the sum, these are the 4 papers claiming reproducibility over time. And the investigator himself says this is a growing body of supporting empirical evidence for direct effects of conscious attention of human participants on photon interference pattern in a double-slit apparatus. And of course, as you can imagine, enormous interest in this, millions of different videos achieving millions of YouTube hits, 2 books on it, interviews. Next to the BEM study, I think it's the biggest other study that gets recognition in the community. And so what we did is we approached Dean Radin. And so it might even be a theory behind it. I remember quantum theory is responsible for many strange things. And if you always, when you need to explain something strange, you use the quantum. And so in this case too, the most popular mechanism for anomalous matter interaction is based on quantum mechanics, particular proposal that observer consciousness collapses the quantum wave function, which is in line with the Copenhagen interpretation of Heisenberg, Wigner even, people, played with that idea. But, even Abner Shimoni, one of the leading, thinkers in quantum theory, in the 70s, did that experiment and published in Foundation of Physics. This was not totally, crazy. This was in the 70s thought to be a viable mechanism. So what happened? You have an interference pattern, and somehow the mind. And when you observe, I'm just going to show these other examples. This is from Radin himself. You have intention on the double slit. Somehow you collapse the wave function, have subtle effects. Crazy, yes, but independent of the theory, you can test it in the lab with a real experiment. So the experimental condition is just the condition concentrate, told to the test subject, and the control condition is withdraw attention, just relax. So, and that should reduce the intensity at peak interference of the interference patterns. So there's a camera readout, it's a laser, there's a CCD camera capturing the image, then you have a recording out of that camera, and basically this is in time, and then you have at 30 seconds interval concentrate, and then the experimental intervention, and then 30 seconds for the control condition, just relax. And there should then be a difference between these two after you've done a huge number of experiments and repeat this. So what we've done is we've approached Dean Radin and we found him to be cooperative and commissioned a replication study. And this is really unique. This is why I was excited to present this to you. This is almost an ideal circumstance. The original investigator agreed to perform the study using a blinded protocol with this AMP protocol, using fully confirmatory procedures and pre-specified statistical analysis. The original investigator did it in his lab with the people he selected, The pre-specified protocol was stored on a secure server and was password protected, with the password being in possession of the funder. And of course, as we heard, Soltman, there's other problems with that. You can never be sure. But this is how we've done it. And then the independent data analysis was done in Berlin at our little think tank by having a digital copy of all the 10,000 test trial data, the methods in the form of the original MATLAB software script, and the analysis performed by us. So this is how it's done. There's a 20-minute session, one test subject. The computer gives instructions, now concentrate, now relax, in the presence of this device. And one session consists of 5 of these XO experiments at randomized temporal positions. For each run, it's totally randomized, when it happens, when it doesn't happen. And so this is with the test subject. These are the conditions. And then you have 250 test sessions, giving you about 1,250 only for this condition. Now, the counterfactual meta-experiment, you do the same thing without the test subject. Just logic, just very plain, experimental logic. And that gives you this whenever you've asked, these are the labels for the sham experiments. And what you do is you mimic the random positions of the real experiment, of the true experiment. So you really just mimic as closely as possible to have a counterfactual experiment. Counterfactual experiments, you can never do the experiment for real because counterfactual experimentation is impossible. The universe has moved on, things have changed. But you can mimic as closely as possible. And that's what we've done here. So you're collecting these experiments, the same amount of data for these things, and then in the end, you have a true condition and a sham condition. And the prediction would be, of course, in the true condition, and predicted was a decrease effect of this. This is a statistical significance hitting. And of course, in a sham condition, you shouldn't see anything because there's no test subject, there is no psi, nothing should happen. So there's 4 possible outcomes using counterfactual meta-experimentation in this experiment. This will be a confirmation of the study. Yes, there's a clear predicted effect in the true experiment, no effect when the test subject is absent, so it worked. Or there's no artifact, but there's also no effect. And these are the cases where actually you have an artifact because you have statistical significance when you do the counterfactual run in the absence of test subjects. And that's what we term Class A error when you detect that. Now, what is Class B error detection? I'm going to give you a summary of the results. How much time? Not much time. The true experiment, the systematic negative control experiment, is to do a relax versus a relaxed experiment. Because what might happen is that the first control might bias the second control, these 2 conditions. So you want to see whether there's statistical error with that. Done in the same amount of data. Then also you've done it for the concentrate versus concentrate conditions, which we call systematic positive controls. Then you do it for let me just see, then you do it for other way. Time reversed controls, you do the control before the concentrate case. And in the end, you have all these different random positions with differently randomly assigned experiments, and they combine in a full experimental set. And for each 20-minute sessions, of which were done 250 over several months, each time the whole thing is randomized in a different way. And of course, you also do this for the sham experiment. You mimic that. So this is a total of 5,000 test trials. Oh, it's time up. Good. Let me just rush to the result. 5,000 test trials. I'm just going to skip the definitions of these and come to the data. These are 8 different null hypotheses, by the way, with their own dedicated data sets. And only these 2 show real results, and these 2 are only done to detect errors in the system. So this is what happened. The actual experiment, after unblinding, after 1 year of collecting data, showed none of the predicted effects happened in a true experiment when the test subjects were present. But in the counterfactual case, when test subjects were absent, one of the test conditions showed a highly significant, statistically significant effect in a predicted direction, in a predicted position. So what this shows is that an artifact can clearly happen in a system when you run this experiment, even without test subjects reaching significance. So this is the overview of the data. Remember, when you do an experiment, you only run the standard experiment, the XO condition. We've added 7 different categories of experiments to track different error sources in the system. if you would not have done that and just run the experiment, and it happened to be this one, where there was no test subject, there was a 50% hit rate, and it would have been interpreted, oh, there's ESP, or there's a PK effect in the system, when there really wasn't. So, and then you can actually quantify the amount of error that you have in there. This was just published on August 22nd. And I'm proud to, and here's the interesting thing, the reviewers, this in Frontiers of Psychology, the referees, it was a long review process, but in the end, these two referees, Markus Meyer at the University of Munich and Harald Wallach, they're well known to, they're supporters of psi research. They have published in the past psi research. And so not skeptics reviewed the paper, but in fact proponents reviewed the paper and basically agreed with us that there was a false positive effect in this design that was most likely responsible for disclaimed reports. And so I finish up 2 minutes, if I may, John. The poster that was presented then took the next step. And this was the work performed by Nikolaus von Stilfried, who is here, who you should talk to, in Berlin. He asked the question, could the prior results then of all the previously published studies have been uncontrolled false positives as a function simply of statistical errors? And so the project was to collect 8 times the amount of data produced in the commissioned replication study just on the apparatus with the same protocol, but of course there was no psi present at all. And then he was implementing the same MATLAB scripts for statistical analysis that were approved by Dean Radin from his earlier publications. And what he found was that the data analysis with the empty experiment got a lot of statistically significances at different positions for these 64 data sets. And he identified that the problem was a trimming function in this particular case, an overly restrictive outlier removal procedure. And when he switched off the trimming function, then it was just chance expectation events in the system. And when the trimming function was on, there were just highly statistically significant false positives all over the place. So the conclusion was that the trimming procedure, which is used by Radin et al., can lead to false positive results. So the reason I'm telling you this is, with a sham experiment performing this counterfactual meta-experiment, if you run it, you can detect false positives no matter what their origins are, no matter what its source is, whether the error might be a statistical error, whether the error might be a wrong sample, whether the error might be a misapplication of control conditions. It doesn't matter what the source is of the error in the system, the counterfactual meta-experiment will pick it up. And so what I'm ending up with is this slide. So I'm going to skip this. So this is my final slide. So in the past, certainly in parapsychology, but also in anomalies research in general, where people are pushing the boundaries of scientific inquiry towards new phenomena, for breakthrough phenomena, I think the question was, until now, can experiment be replicated? Can it replicate the original finding or not? I hope in the future, by adopting protocols like this, we can ask the question, can the experiment show that false positives are absent in the case so-called true positives have appeared? And I think this will be a major advance at the experimental level of the scientific process, what everybody can do on any particular sort of experiment, either in physics, biology, chemistry, psychology, wherever you go. And so I just ask, amplify the reliability of your research design. Look at this paper. We had enormous responses to this. I mean, there were 300 retweets already in the past 2 weeks. There were, 2,000 views almost now, just in 10 days of this paper. So I think there's a need for new research protocols. Beyond just going beyond pre-registration, beyond doing your statistics, actually do measure the error in your system directly. Thank you very much. Please go ahead.
Hi, Joshua Walleczek, Yale School of Public Health, meta researcher. Fascinating talk. I have a question. So you say that this could be used in all experimental settings. So in the clinical medical world, you gave an example of intervention, aspirin, placebo, sham. So in that case, is the measurement that would be taken on no subject at all? So if you're measuring like blood pressure,
Very good point. I mean, this can be, the adaptation of the protocol is at the beginning not very obvious in talking to people. But what this would mean is there would be a patient, but he would receive no pill. And then you would have a patient receiving a placebo pill. And then you could compare what is the placebo effect directly in this case. So the moment the outcome variable, y, is generated by the subject, him or herself, you need to change the protocol in some ways. Here it's a physics experiment where you just have a single outcome variable, and then you can controllably apply all those different conditions. So because the outcome variable is not generated by an endpoint coming from a human test subject itself. But it can be adapted, and you need to look at the case by case what would be the best case. So at the minimum, I would say, for many, so in clinical trials, we do some of those things already with placebo controls. That's why we invented placebo controls. Right. Yeah. So my early ideas came from, I worked at the Stanford Medical Center for 8 years, and so I was very, looked at the trials, and out of these, I got these ideas on how to do experiments better when we compare insights, originally coming from clinical research to biophysical experiments. And all I'm saying is it can be adapted, but it's a different process. This would be a different protocol than what I've shown you. But the basic idea of doing counterfactual meta-experimentation does work in these cases as well. Thank you. Please.
Yes, thank you. Yeah, I do very much like the idea of counterfactual experimentation with sham data. That said, do you actually need a separate sham group for that? I mean, maybe I'm missing something, but I mean, couldn't you do more or less the same thing in terms of like just testing out the code and seeing that a lot of false positives come up just by shuffling the data or using like control versus control?
No, what we want to do, exactly, your last point, control versus control, right? If you run systematic negative controls, you're actually looking only, you have the source. When you do a control, the source of, the source generating your experimental condition is also the source generating your control condition in most experiments. Now in the sham experiment, that source is gone. So when you do a control versus control, you still have the source present that generates your control. In the true sham experiment, so the double S, sham versus sham, that source is gone altogether. So the systematic negative control is therefore measuring a different class of errors, which we call class B error, which is a condition-dependent error. And the sham versus sham is measuring a method-dependent error, independent of the application of control and experimental conditions. So they really look at the paper. It's not very intuitive at the beginning, but it makes a lot of sense once you get a head around, because we're not used to thinking this way. But good point.
Hi, thanks. Jessica Logan from Ohio State University, and I am a quantitative researcher and methodologist. And I'm wondering if you can talk a little bit about, I think you started to get into it there, but that feels a lot like sort of between subjects and within subjects, typical experiment that I might see within any sort of social science. So the idea, it sounds like, is there's a between within a segment effect versus a between segment effect, and sometimes you have a person present and sometimes not. So I think the question is just, it felt very typical, sort of like an experiment I would see all the time in a social science research journal. Can you talk a little bit more about how it's different?
Well, outside the biophysical domain, we are now testing it, starting to test it in cognitive psychology. But social psychology is a whole other domain. And in my view, I don't quite know how you run experiments where you have the control and the experimental condition completely absent in social psychology experiments. So you have to help me out on that. Because usually you look at data that already exist and to analyze in a certain way. But actually to do predictive experiments on these outcomes, that's a very different animal altogether. And I think there, you really have to think about what would be a sham condition in that particular case.
So sometimes we have an active and a passive control. So if I'm trying to improve children's reading ability, right, I might give them a reading intervention as my active control. A passive control would be I give them a math intervention. I don't expect the math intervention to improve them, but some of the other mechanisms are there. I'm there working with a student one-on-one for a long time. but then the control condition is a lack completely of that experimentation. And so if you have a within subjects and a between subjects component, am I getting at the same basic process, or do you still think it's different?
What I would suggest in this particular example, you can actually, with this protocol, if you adapt it to this situation, you can actually measure any error that might be associated with all those different conditions that you're applying, the active and the passive. And usually you don't have that information. So we can talk about that.
Okay.
Yep. Zoltan.
Thank you very much for the wonderful talk. Zoltan Kekec, psychologist. Yeah. So I'm interested in how you powered your study, because that was one of the other extremely difficult case in our case, how to define the minimally interesting effect, because in parapsychology, you can make the claim that any size of an effect could be interesting. So how did you go about powering?
So this was easy in this particular case, because what he said is we focused, and I skipped that slide, we focused on the original study of 2012. Then that Radin claimed he subsequently replicated, certainly with exploratory measures, or not always conceptual, not always confirmatory. But the original study had exactly 250 test sessions and 5,000 test trials. So we just used exactly that experiment, and just, of course, had to double the test trials because we added the duplicate sham condition.
Yeah.
And that's how that was defined in this particular case.
Yeah, and just one note that don't know if you're aware of, but the LAC, so the Atlas project, they have sham data, and also in the gravitational wave detection project. So in these large physics experiments, they are using sham data and sham experiments.
But exactly, in the big physics experiment, I think look at CERN or in other places. I mean, you have whole statistics departments basically just looking for null effects and doing all your simulations based on negative controls and null effects. But typically in our psi experiments or cognitive psychology experiments or at the limits of resolution, when they need to do that because they understand, they're looking at effects, one part in 10,000 or one in 1,000 sort of parts per million effects, and then you need to do that. And we normally, as lab researchers, in biology, biophysics, or psychology, we're looking for bigger effects. But once we push our experiments to smaller and smaller effect sizes, we still use the same tools and just say, oh, it's all going to be resolved by increasing my sample size. But this shows you really don't, because other error sources kick in that are not covered by your regular statistical analysis. So that's why I'm saying statistics, there is no statistical solution to this problem. That's why you need to empirically detect those errors and show that they're there or that they're not there. Thank you. I think
we are out of time. I will take the chair's prerogative, though, to say, just in case this isn't completely clear, I think the exciting thing about what Jan has come up with, which took him like 2 hours to explain to me before I finally got it, is that so much of our statistics and science, and by the way, that 2 hours was my fault, not his. The logic of what we normally do statistically is to make assumptions about error structures. And what's innovative and revolutionary here is such a simple move to actually estimate the errors present in your design in that situation with those participants. And so you don't have to make those assumptions anymore. So thank you very much. Coffee break time.
Thank you.